Welcome to today's SCOG lecture. So my name is Peter Tessels and I'll be chairing today's session, and the talk will be given by Ashley Sanders from the Berlin Institute of Health at the Charité. But before I introduce Ashley, I would like to give two announcements. So the first one is for the upcoming lecture in 2 weeks, so on the 22nd, same time, which will be given by Michaela Delady from the DZNN in Tübingen. So please join again. And then the second announcement is for the SCOG virtual workshop on recent advances in single-cell epigenomics. And you can see there's a great lineup of speakers. And although as far as I understand, the abstract deadline is closed, the registration is still open. So please sign up and join for this event. Okay. Cool. So now to Ashley. So Ashley, let me just quickly introduce her. So Ashley is a human geneticist who was trained, I think, in Canada. And then after her PhD in Peter Lansdorp's lab, she moved to Jan Korbes at EMBL. And throughout her career, Ashley has been very much interested in understanding single cells, and particularly structural variation in single cells, and trying to understand how they impact somatic and germ cell biology. And I think this is probably also what you'll be talking about today. And what I should also mention is that Ashley moved to the BIH early this year, and she's, I think, in the process of really establishing her lab. And we are very much looking forward to your talk. Thanks for joining.
Great.
I should also say, sorry, Ashley, sorry, some housekeeping. So the presentation will be roughly 30 minutes. It will be recorded today. And if you would like to ask a question, so there are two options. Please either put them in the chat and I will read it out loud. Or if you like, please raise the hand and then we can unmute you and you can ask the question yourself. All right, perfect. So Ashley, the floor is yours. You have to unmute yourself.
Okay, I think you guys can hear me. Good morning, everyone. Hello. It's so nice to be here, and I really appreciate the invitation to talk to you today. So today I'll be telling you about some of the single-cell multi-omic techniques that we're using to characterize somatic mutations in single cells using my favorite technology, which is StrandSeq. It's single-cell and single-stranded, and I will give you some details about that later. But first, I wanted to take a moment just to introduce myself, as this is the first time I've had a chance to talk to you. So I'm a new junior group leader, as Peter mentioned. I just started my lab earlier this year in Berlin, and I was actually recruited in the single cell focus area for single cell approaches for personalized medicine, which is a focus area that was jointly formed between the Charité, the BIH, and the MDC. And I was recruited along with 3 other great group leaders, my new colleagues, Simon Haas, Leif Ludwig, and Stephanie Grosswand. And together we are all paired with clinical partners here at the Charité. And the idea is that we apply single-cell genomic technologies towards translational research. And we are able to achieve this through this really nice tandem pairing with our clinical partners, because they are able to provide us with key patient materials to apply our most favorite single cell technologies on, and also, of course, make sure that our research questions are really grounded in biomedical questions. So we're very excited about the new single cell focus area. There's a lot of energy here in Berlin around this, and I do hope you will all look out for some new exciting opportunities that are coming out of Berlin now and through our institutes, and including some regular networking events and lecture series that we're currently organizing. So please do keep an eye out for those. So with that, I'll get into the science. And I guess the overarching question that my lab is interested in, and of course a longstanding question is in biology, is understanding how genotypes really change phenotypes or inform different phenotypes. And in order to do this, typically the approach is to detect genetic variants in different human populations, disease cohorts, or even in single cells to explore the genetic variants that are enriched in different populations and try and associate that with specific phenotypes or disease outcomes. Now, in order to be able to really explore these questions, we need reliable tools that map all types of genetic variants in individual genomes. Now, beyond the germline genome, which is really the genome we inherit from our mother and father, somatic mutations also arise in our tissues as we develop and age. These occur in a single cell typically, and the mutation is then inherited by the daughter cells. They can be clonally selected for over time and in fact expand into subclonal populations. And as a result, in any given person, there may in fact be many hundreds or even thousands of tissue-specific single-cell genomes. Understanding how these contribute to the health of our tissues is really an active area of research in our laboratory. Now, somatic mutations have, of course, received a lot of interest recently. It's an emerging field, I would argue. And it's been shown, certainly, that the mutation rate increases as a function of age in the vast majority of our systems, and that some individuals show a very elevated rate of mutations that's associated with their disease. However, the vast majority of this work is really centered on studying the contribution of single nucleotide polymorphisms, so really single base pair changes, and have so far really neglected to explore the contribution of structural variants. Structural variants are larger changes in our genomes that involve copy number gains or losses, or also copy neutral events like inversions or translocations. And these can have large impacts on the biology of our tissues because they can change not only the gene dosage, but also the regulatory environment that's kind of regulating or controlling the expression of genes within these rearranged chromosomes. And a recent pan-cancer survey actually illustrated that 55% of cancer driver mutations are in fact structural variants. And I bring this up because it shows that structural variants actually occur somatically in our cells very frequently, and that they can account for more base pair changes than single nucleotide polymorphisms alone. And these changes can really be driving different disease phenotypes in our tissues. So understanding how somatic structural variants emerge in our different systems when they disrupt key pathways and lead to disease is really the overarching theme for my research in my laboratory. So one of the ways that we are approaching this problem is by using single cell and single strand sequencing. This is a technology that is very close to my heart as I was involved in developing it with my PhD mentor in Vancouver, Peter Lansdorp. And StrandSeq is really a single cell approach that not only sequences the genome of a cell, but it also sequences only one strand of DNA within the genome. And so for that matter, for that reason, I like to consider it as not sequencing a single cell, but actually sequencing half a cell. And I hope that by the end of my talk, you'll see how sequencing half a cell can sometimes give us more information than when you try and sequence the entire cell. So to understand StrandSeq, we first have to recognize that DNA is double-stranded. And what that means is that each strand of a DNA molecule can be distinguished based on its 3' to 5' directionality. We have a Watson strand that I show in orange and a Crick strand that I show in blue. Also, of course, humans are diploid organisms, so we have 2 copies of every chromosome in our genome. One we inherit from our maternal lineage, the other we inherit from our paternal lineage. Whereas standard next-generation sequencing technologies typically ignore this information, StrandSeq really takes advantage of it. So during normal cell division, each strand of the DNA molecule serves as a template for DNA synthesis during S phase. We culture cells in the presence of BRDU for a single round of cell division, so that we generate these hemi-substituted sister chromatids. What that means is that one strand, the dotted line, contains BRDU, and the other strand, which is the solid line, does not contain BRDU. This allows us to selectively remove one of the strands during library preparation so that we really only sequence the other strand. So during mitosis, these sister chromatids segregate into the daughter cells, and it's at this stage that we capture this cell, and we subject it to a modified Illumina-based protocol that really just ligates on these forked adapters onto the DNA fragments. And we do all of this, of course, without any whole genome amplification, because if we performed whole genome amplification, we would really lose the template strand identity, which is so key for the protocol. At this stage, we remove the BRDU strand using a photolytic cleavage event that really nicks the DNA strand at sites of BRDU incorporation, and that leaves only the template strands available for sequencing, which we are then sequence and align to the reference genome and are able to distinguish the directionality of the template strands based on whether they align to the plus or the minus strand of the reference genome. And so from a single strand-seq library, we can immediately see every single template strand that was inherited in this daughter cell. We can identify when chromosomes were inherited in a Watson-Watson orientation. So both of the homologs were inherited as a Watson identity, or chromosomes that were inherited in Crick-Crick identity. So both homologs were in the Crick orientation, and chromosomes which are inherited in this Watson-Crick combined configuration. So one homolog was in Watson and the other homolog was in Crick orientation. And because each template strand really represents an individual homolog, we can use this information to directly track the individual chromosomes within a cell. So if we look at a pair of sister cells, we can see reciprocal inheritance pattern for every single chromosome. And I really love to show this slide because I believe it really highlights that the template strand segregation pattern is directly tracking the individual homologs within a cell. Here, a single cell was sorted, allowed to divide in vitro, and then I manually went in and picked out each of those individual cells, and I generated strand-seq libraries from them. And you can see that if sister cell A inherited Watson-Watson, sister cell B inherited Crick-Crick. If sister cell A inherited Crick-Crick, sister cell B inherited Watson-Watson. And if sister cell A inherited Watson-Crick, sister cell B inherited Crick-Watson. So this mirror image template strand segregation pattern really points out that each strand represents an individual homolog, and that by sequencing just the template strands, we are in fact preserving homolog identity. And it's really this feature that separates strand-seq from all the other sequencing technologies in the world. By sequencing just template strands, we're able to extract more information per genomic locus. For instance, for any given genomic region, we can actually combine 3 distinct data channels to predict structural variants at the genomic position. And these include your depth, so the total number of reads that are present in the genomic locus, the strand, so the proportion of Watson or Crick reads, and the phase, which is the number of reads represented by either the maternal or the paternal homologs. For a reference state, we would imagine that we would have a 2n depth of coverage. We could have both of the strands being equally represented, and of course we would have both homologs present at that genomic position. This is distinct from a deletion, where we would have a reduction in the read depth signal. We would have only one strand being represented, and we would have a complete loss of the haplotype or the phase SNPs for the affected homolog. And this is distinct from a copy neutral event, such as an inversion, where, of course, because it's copy neutral, we don't see any change in the read depth. Instead, we see a shift in the strand orientation, and also all of the SNPs that were present on the inverted allele are now flipped in orientation as well. So by integrating these 3 data channels, we're actually able to distinguish a vast majority of different structural variation classes. And in fact, we've described diagnostic footprints for several disease-relevant structural variants. These include, of course, your copy neutral changes, copy number changes, copy neutral changes, such as inversions and translocations, more complex rearrangements like inverted duplications, copy number LOH, and also chromosomal aneuploidies, or even whole genome doublings. And the key point is that the vast majority of these are actually not possible to resolve using many other sequence-based approaches. And so these diagnostic footprints really gain us access to a larger spectrum of structural variants than previously possible. So to show you how this looks in real data, I show you here an example of a deletion that we located on chromosome 3. And if we dissect out this deletion into those 3 data channels, we can see the depth, strand, and phase all show the stereotypical changes in their data channel, which we predicted based on our diagnostic footprint. So we have a reduction in the depth, only the crick strand is represented, and we have a complete loss of the H2 SNPs that are represented by the haplotype 2 of that homolog. We can see a similar pattern for a duplication, or the reciprocal pattern for the duplication, along with an inversion and an inverted duplication. So really all of these loci represent the patterns that we were predicted to see based on our diagnostic footprints. And what I really just want to draw your attention to is that the information or the data that's present in any given data channel is actually quite subtle. So the signal is quite subtle in the depth channel, for instance, of this duplication, this 1.5 duplication, megabase duplication on haplotype 1. It's really by combining information across these 3 data channels that we're able to bolster support for the structural variant and make a more accurate call about what's happening at that genomic position. So to do this systematically, we next developed a computational tool with Sasha Myers and Miriam Garagani, who was at the MPI in Saarbrücken. And this tool uses SC-TRIP, which is single-cell tri-channel processing, to locate structural variants systematically using a Bayesian framework. How this works is that from a single sequenced strand-seq cell, we segment the single-cell data to assign the template strands for each single bin, also to phase all of the haplotype SNPs, and also to count the proportion of Watson and Crick reads in each of the defined segments. We then compute the likelihood for each of the diagnostic footprints we described in a haplotype-aware manner, so that we're able to assign a probability score for every possible structural variation footprint, and then classify the locus based on the most probable structural variation class. And the output of this is really a list of structural variants for each single cell with probability scores attached. This really allows us to map structural variants across an individual cell, and also gives us an opportunity to explore the subclonal structure of the sample. So we can visualize these data as a heat map, and that's shown here, where we have all of our cells present on one axis and the structural variants on the horizontal axis, and the color of the pixel really represents the most likely structural variation class that we predicted for that genomic location. We extensively benchmarked our workflow and found that we identified structural variants as small as 200 kb in size, which is really up to 50-fold greater precision or resolution than other techniques offer for structural variation detection. And we also validated 97% of our high-frequency events by bulk analysis, by really deep sequencing of bulk cells. So this opportunity to map structural variants in individual cells allows us to immediately identify subclonal populations within our sample to explore how genomes evolve in our systems. And to be clear, this is really several orders of magnitude better resolution than the current state of the art. Most efforts to study structural variants in single-cell data use single-cell RNA-seq data to predict copy number variants, but really they're limited to only detecting these events on the chromosome arm level. And that's because you need so many reads to really identify the copy number variant. And so using strand-seq, we're not only identifying smaller SVs, but also a larger range of structural variation classes that can have large impacts on the biology of our systems. So what that means is that we now have a robust method to detect a diverse range of structural variants in a cell-by-cell manner and explore the evolution of a system in a clone-by-clone manner. We can use this, for instance, to identify low-frequency genomic rearrangements that are present in a patient sample. Here I'm showing you the heat map for all of the chromosomes of a strand-seq experiment performed for a leukemia-derived sample, so a patient-derived xenograft sample from a leukemia relapse. And when we looked at the heat map, we identified, of course, a clonal rearrangement on chromosome 7, where we can see all of the cells have a deletion of one arm and some complex duplications on the Q arm of chromosome 7. But we also identified this subclonal population here on chromosome 6. And when we zoomed in to cells that had this pattern of structural variants on the Q arm of chromosome 6, we actually identified 13 distinct breakpoints, all residing on a single haplotype, the haplotype 2 of this chromosome. And when we then looked at the SC trip signal for this genomic region, and that enabled us to very finely define the genetic rearrangement process that happened on this haplotype. So we identified several deletions, inversions, a duplication, and a large inverted duplication. And once again, these really all resided on the single haplotype 2 of this chromosomal arm. Having these breakpoints available to us, we then predicted that this arose through a chromothripsis event. So chromothripsis occurs when a chromosome becomes severely destabilized and the actual homologue becomes shattered, often during a mitosis. And then these individual chromosomal segments become glued back together in a brand new configuration. And so this is often results in inversions and segments of the chromosome being lost, which is really very similar to what we predicted for this genomic locus. So we predicted that a single chromothripsis event arose in the relapse, and then there was a selection event that enabled it to increase to the frequency of 31% within this sample. Having these breakpoints very finely mapped by using the signals in our single cell data, we then looked to see if we can find evidence of the chromothripsis event by doing very deep bulk sequencing of over 160x. And without knowing where to look, we did not see evidence of the chromothripsis event, so it was missed in this deep sequence data. But thanks to knowing exactly where we predicted the breakpoints to be, we were in fact able to find these paired read signatures that validated the reconfigured chromosome that we predicted from our chromothripsis event. So really this shows that StrandSeq provides a non-targeted opportunity to explore the genomic rearrangements that are arising in a patient sample. And once we have a prediction of what happened at that chromosome, we can use the breakpoints that we've identified to get more resolution of the event using other sequencing approaches. So what I think I've shown you so far is how single cell sequencing can really uncover more genetic variation than previously possible. We can also use this to explore genomic variation within a germline. And one example of how we're exploring this or how we're using these data is to combine the structural variation predictions for each individual cell in order to generate these high resolution and haplotype-resolved digital karyotypes. So in this example, we generated StrandSeq libraries for another leukemia patient, and then we used a single cell SC trip to build the first ever digital karyotype by finding the consensus karyotype across the single cell populations. And what we found for this individual is that they had Klinefelter syndrome, so an extra copy of chromosome X. They had trisomies for chromosomes 7, 8, and 9, several hundreds of megabases of loss of heterozygosity that was copy neutral, focal deletions and inversions that affected known tumor oncogenes, and also a cryptic translocation that resulted in 2 derivative chromosomes involving chromosome 5 and 14, and that really resulted in a rearrangement product that placed the TLX3 oncogene near a BCL11B enhancer region. And so this level of resolution that we're getting for this individual patient sample was really coming from just 41 single-strand seq cells. And if we calculated the cumulative coverage across all those 41 cells, we have less than 1x coverage. And so we're really getting very high resolution and sequence-based karyotypes for an individual genome with very little sequence input. And to give you a little bit of context, the current state of care is still to generate these 2-dimensional flat maps or cytogenetic karyotypes, which uses banding pattern along a chromosome to predict genomic rearrangements and copy number changes, etc., using cytogenetic karyotyping. And so I hope you can imagine how transformative sequence-based digital karyotypes can be to improve our ability to diagnose patients and to better resolve the genomic configuration of different disease systems and cohorts. So being able to locate more genetic variation actually gives us better insight into patients' diseases. As I mentioned, there was a translocation that we identified between chromosomes 14 and 5 within this leukemia patient, and it was cryptic, meaning that it wasn't visible in the cytogenetic karyotype. When we explored the breakpoints of the translocation, we predicted one of the breakpoints were just 3' of the BCL11B enhancer regions, and upstream, the other breakpoint on chromosome 5 was very near to the TLX3 oncogene. That indeed resulted in a derivative chromosome that would have placed the BCL11B enhancers just upstream of this oncogene. And we predicted that the translocation would result or enable an enhancer hijacking mechanism by enabling the BCL11B enhancers to drive the expression of TLX3. And indeed, when we looked at the RNA sequencing data for this patient sample, we found that TLX3 was vastly overexpressed within this individual. This really highlights how mapping genomic rearrangements in patient samples can allow us to better understand the mechanisms that are driving their disease. And having a higher resolution picture of genomes is very important for making more informed decisions about how to treat them, because now we know the driver mutation that really was responsible for this individual's leukemia. So mapping genetic variation is, of course, one part of the puzzle, I suppose, but identifying or predicting the consequences of these genetic mutations is really critical for making that genotype to phenotype connection. And some of the work that we're doing now to uncover this is really using the nucleosome occupancy signature that's available in our data to make predictions about the functional outcomes of structural variants. We're able to do this because StrandSeq uses MNAs to digest chromatin, which cuts the DNA between these nucleosomes. And as we can see, if we look at the read depth plots of a StrandSeq library, we can see that the depth very nicely mirrors that of an MNA sequencing experiment for a bulk sample. And that's because we're really chopping at 150 base pairs between these nucleosomes. What's more is that we have the haplotype information available, so we can also dissect out or separate the sequencing data based on their haplotype identity, giving us an opportunity to explore allele-specific changes in nucleosome occupancy within our single cell data. Because gene expression remodels the chromatin and expressed genes really are depleted of nucleosomes, we can use the MNA's digestion pattern as a readout to predict the expression level of different genes. And we can see that there's a negative correlation between nucleosome occupancy and gene expression, where we have a depletion of nucleosomes for the highly expressed genes shown in red, and we have overrepresentation of nucleosomes or higher read depth for non-expressed genes shown in gray, for instance. And indeed, we see these dips at the transcription start and end sites as would be expected for these types of data. Using nucleosome occupancy to make predictions about the global expression pattern of genes gives us the new opportunity to use these data to not only predict the cell type identity based on global expression patterns and differences between individual cells, but also to make predictions about pathways that are disrupted in clones that contain a structural variant versus clones that do not. So we're really using the nucleosome occupancy to make predictions about different signaling pathways in a clone-by-clone manner. To show you how this is working, we applied our approach to that chromothriptic clone that I described previously for the leukemia patient sample. And within this chromosome 6 Q arm, where we identified the 90 megabase chromothripsis event, we see multiple transcription factors that are present within this region. If we look at the nucleosome occupancy of these transcription factors, we indeed see a haplotype specific differences between the level of coverage at these transcription factors, and a difference between cells that harbor the chromothripsis event versus the major clone, which is really your wild type or the normal cell population. A transcription target factor analysis revealed that MIB was the most differentially regulated transcription factor that is specific to the chromothriptic clone. And indeed, MIB is a master regulator of many important genes that control the proliferation and survival of cells, including Notch1, a well-known oncogene. This suggests that the chromothriptic clone enables overexpression of MIB, driving Notch1 activation within cells containing the chromothripsis rearrangement. And that means that if we now target Notch, because there's many drugs that are available that can inhibit Notch, we might be able to specifically inhibit the chromothriptic clone in order to ablate this clone from the patient sample. And indeed, our collaborators in Zurich, BIH Bornhauser, is in the process of running those experiments now. So in summary, what I hope I've been able to show you is how we can use single cell and single strand sequencing to uncover a variety of disease relevant and very important structural variation classes in individual cells. And we have robust computational pipelines now available to build high-resolution karyotypes that allow us to explore the genetic variation that was inherited across the population of cells. Also to look at the individual variants per individual cell to really explore the mutational landscape of structural variants within a subclonal population. And then use the nucleosome occupancy to simultaneously read out the pathways that are disrupted in different subclones of our population. And what I think is quite powerful from this is that we're extracting, or we're getting all of this data from really a single sequencing experiment, allowing us to really link the relationship between germline variants, subclonal mutations, and aberrant pathways from a single sequencing experiment. So with that, I would like to thank, of course, everyone that's helped generate some of these data. My previous mentor, Jan Korbel. I hope I already highlighted Hubin, Sasha, Karen, David Perupsky, and Miriam for their great work. And also my new team here in Berlin. We're a new group that are just growing, and we are very happy to see where this takes us, because now we have the really fantastic tools to begin applying this to patient samples in order to explore how structural variants emerge in different disease contexts. And with that, I will thank you for your attention and take any questions if you have some.
Thank you, Ashley. This was super cool. Thanks. There's already a question. So so for everyone, just as a reminder, please raise your hand or put the question in the chat. Maturin, let me see if I can.
Yeah, can you hear me? Yeah, I actually, yeah, that's Peter said, very impressive. I was wondering, so you need one cell division event, right, to get this strand. So for now, did you try to have an overview on which kind of tumor you can use that on?
Yeah, this is a great question. So certainly we need a single cell division in order to label the DNA strands, and I really haven't found a better way to label them yet, but I'm happy to explore that if anyone has ideas. But many cells will divide at least a single time, right? Because we really don't need to expand the culture, we just need one cell division. But with that said, solid tumors are very reluctant to culturing, which I find quite counterintuitive because they grow so well inside of us, but then they really hate to grow in a dish. So
I was wondering actually if you started exploring the potential with organoids, right? Because that's really what brought some new way of culturing some solid tumor exactly.
Sorry, I missed the...
Did you start looking at organoids?
Oh,
So
yes.
Yeah, exactly. They don't grow well in a dish, but some of them we now know how to grow it really, at least in a dish.
Yeah, so we certainly are developing organoid systems already and there's many opportunities to explore different tissues using organoids that I'm very excited to be able to use, including, of course, the brain and the gut and neuromuscular junction, etc. So I do think that their organoids provide us a great opportunity to apply this to more tissue types. And really just getting the cells to divide a single time in vitro, you know, is often achievable, but I know that not every tissue is available for us to explore this way, of course. Yeah, but organoids are a great answer for that, indeed. Thanks.
So actually, I was intrigued by, especially by this nucleosome profile. I thought this was very smart to exploit this, but the question is, can you even get more information also? Is there a correlation, let's say, between nucleosome occupancy and the frequency of structural variations that you see?
So we're using the nucleosome occupancy certainly as a readout of the epigenetic profile. And then we're using the SCOG, the structural variants, to correlate changes in the genome to changes in the epigenome. And certainly we are seeing a correlation between the two. What's really nice is that we get haplotype information, so we can directly look at the same homolog that has a genomic rearrangement and ask what are the effects on the nucleosome occupancy within the local region, so cis effects. And we can also use the nucleosome occupancy across the genome to look at trans effects. And that's actually where we're seeing more of the effects and more global changes in transcription factor profiling or in entire regulatory networks that are disrupted based on these structural variants, the subclonal events, really. The power there is to collapse the cells based on their clonal composition, by using the structural variation profile, so that we can get more in-depth information about the occupancy or the epigenetic profile within each clone.
And if I may, so you talked a lot about the resolution, right? And you see, and I understand the enhancer, so that's fine. But how do you really can pinpoint the breakpoint? So because you know a lot of in leukemia, you often have these transcription factor fusions and so on that drive then the cancer. Do you see them as well?
So certainly, breakpoint resolution is not the most precise, I should say. It really depends on the frequency of the event. If for every cell that contains the genetic mutation, we can collapse the reads across the cells to get closer predictions on the breakpoint, but we're certainly not getting base pair resolution. If for every single cell, we're getting about 0.1x coverage. So we're really sequencing only 1% of the genome, and we don't have every single read or genomic position covered at all. Instead, we sacrifice coverage for this long range information. Yeah. So when an event is very common or clonal within the population, we have seen quite precise breakpoints. But again, it's really dependent on how deep the sequencing reads are within that genomic position. Yeah, but nevertheless, we are able to make some pretty good predictions about the overall regulatory environment, and even have seen changes in TAD structure using the nucleosome occupancy that region. So we're really kind of exploring how deep we can get with these cells or these samples now.
So there's a question in the chat from Benji Tan. So hi, thank you for the nice talk. I was wondering if you can visualize changes in the strand caused by external DNA insertions. So for instance, retroviral DNA integrations by HIV.
Yeah, interesting question. I think, I don't know, actually. So I guess the strand information is really a reflection of the mother cell, because we're tracking the homologs that were inherited in the mother. So the integration site would have to be certainly preceding the cell that we're looking at. But I'm not sure what it would look like. It would really depend, I think. It's a good question, though. I should consider that more.
Okay.
Thank you.
Okay, so there's a thank you. Do we have more questions for Ashley in the chat, or if someone wants to ask? So if that is not the case, I would like to thank Ashley again for a fascinating talk, and I hope to see you all in 2 weeks. Thank you.
Thank you, everyone, and have a great rest of your day, Peter.